for diabetes on the horizon. Also, clotting factors. Does anyone remember the genetic disease that had hemophilia? Very good. Now these people have to take pills that have the clotting factors in them. But if we can produce a bacteria that produces clotting factors naturally, when I say naturally, I use quotes, naturally in their body, then they don't have to take pills. Their body's going to do it for them. So these microorganisms can be very, very important. All right, transgenic animals. Transgenic animals have been used to study genes and improve the food supply. Has anybody in here ever had food poisoning from eating chicken? Yeah. Okay. And how was that, guys? It was not fun. I've had it, and I literally, it was probably one of the worst experiences of my life. I was sweating profusely, throwing up. I was kind of like hallucinating. And basically what happens when you have food poisoning is your body is being poisoned by bacteria. And your body wants to get rid of it at any cost. So it's going to sweat it out, or it's going to come out both ends. Okay. All right. So I got mine from eating chicken. I had a chicken Caesar wrap when I was in college. I never had one again, just because, you know, it's kind of like a mind thing. But what they can do is they can, on the next slide you'll hear about how they can actually produce chickens that are resistant to salmonella, or have them produced with human genes that make their immune systems act similarly to those of humans. And the way that this is beneficial is if the mice have a human immune system that is beneficial to, that acts like humans, they can test cancer treatments, they can look for cures for certain diseases and see how those cures are going to act on the humans even before they try anything on humans. A lot of people have a problem with this though, because they say it's inhumane. So there's a debate on that. And then I just told you about the researchers that are trying to produce transgenic chickens that will be resistant to bacterial infections that can cause food poisoning like salmonella. Okay, they're working on that. And then transgenic plants are now an important part of our food supply. Many of these plants contain a gene that produces a natural insecticide. So plants don't have to be sprayed with pesticides. So they're going to produce that internally. A lot of times when you get fruits and vegetables home, you need to wash them off because they may have those pesticides on them. But these fruits and vegetables are going to actually have them inside. Okay, they're going to be producing. Now, some people say that they don't like that. What am I putting into my body? Organic food is the opposite of this. It's not treated with anything. It's completely natural. All right, now, cloning. Cloning is where we're going to end today. Okay, I'm going to tell you how an organism is cloned. A clone is a member of a population of genetically identical cells produced from a single cell. It's exactly the same DNA as the parent. When you were born, you're a mix of your mom and your dad. Okay, some people say, oh, you look like your dad or you act just like your mom, but you're still a mix. A clone is genetically identical to that one parent. It's only one parent. Okay, I'll give you a minute to take this down, and then I'm going to explain the diagram. It's a very simple diagram. It looks like it's like, ooh, crazy, but it's not. So I'll explain that in a second. So today, after we're done with this, we're going to do a virtual lab. Okay, you'll do some actual virtual gel for electrophoresis. We may not get to that point today, but you're going to do some research on cloning and DNA extractions, and then whatever we don't finish up today, we'll take to the next, when I see you next, which I think will be probably next Wednesday. All right. In 1997, Ian Wilmot cloned the first mammal. Okay, I should say like the first huge mammal. And sheep aren't huge, but they're pretty big organisms. And he cloned a sheep called Dolly. How you doing, Brian?
Good.
Okay. All right, now, this is your diagram of cloning. Okay? What you need to first do is you need to take a donor cell. So for example, if I wanted to clone myself, I would take a normal cell from my body and I would take out the nucleus. Alright? So in this case, it's taken from a sheep's udder. Doesn't matter if the sheep that's donating the cell is going to be male or female. Then you take out an egg cell. The reason why you take out an egg cell is there needs to be a signal that basically fertilization or a combination of genetic material is going to happen, and that comes from an egg cell. Okay? So an egg cell is taken from the adult female. We take the nucleus out. Why would we take the nucleus out and say we don't need that? Megan?
Get rid of the other person's DNA.
Yeah. If you added these two DNA samples together, you're going to get more than the chromosomes that you need. So you want to take this nucleus out and basically say goodbye. All right? So you're going to take the egg cell without the nucleus, and you're going to take your donor nucleus, and you're going to fuse them together with an electrical shock. And that electrical shock acts as the fertilization between the egg and the sperm, even though there's no egg and sperm. Okay? What that fused cell is now going to receive is a signal to start dividing, and it starts dividing into an embryo. The embryo, once it reaches a certain point, is then taken and put into a surrogate mom. Okay, she's going to house the baby for the next however many months. And then after that, little Dolly's born. Okay? So what we have is we have a donor cell, which is going to contain all the DNA of the clone. We need an egg cell, but we don't need its genetic information. We're going to shock it. We're going to shock it with an electrical shock, and once that fusion comes into play, it's going to start dividing. Okay? The embryo is then going to be put into the surrogate mom. She's going to carry the baby, even though it may not have any relation to her whatsoever. She's just carrying it, and then Dolly is born. Okay? Now, this process over here is much like in vitro fertilization. When women can't get pregnant naturally, they go to fertility specialists, and they actually have the fusion of the egg and the sperm done outside the body, and they have the embryo start to grow, and then once they get to a certain point, those embryos are inserted into the woman, and she hopefully has one of those embryos takes, and she can have a baby. But the cloning is just the one genetic information. So who is Dolly resembling here? Which sheep? 1, 2, or 3?
3.
3. What's the sex of a Dolly going to be? 1, 2, or 3? 3. Okay. These two others are just donating things that have nothing to do with the genetic information. Okay. Any questions? Yes.
This is like back to